Hey guys, my name is Kaylee Broom, and today I'm excited to talk to you about plant and animal mitosis and meiosis. So, we're going to be focusing on cellular division in plants and animals. So, first I'm going to talk to you about mitosis, but mitosis can't take place without the cell cycle. The cell cycle happens in three phases: interphase, mitosis, and cytokinesis. So let's take a look at interphase. Within interphase, you're going to have this chromatin, which is uncoiled stringy DNA. And this chromatin can be found in both plants and animals. We see that all these dark spots within the nucleus of this animal cell and in this plant cell is chromatin. So within interphase, all that chromatin, all that DNA is going to replicate through DNA replication. And once that happens, we can move on to mitosis. And mitosis is the division of somatic cells into two genetically identical cells. So say that this image of this cute little doggy right here is a cell. That cell is gonna divide into two genetically identical cells, so two clones of itself. So why is it important that this happens? Well, it's important because mitosis helps us grow, helps us develop, and it helps us to heal. So say you fall and scrape your knee. Well, that's going to heal because of mitosis. Or say you grow from being a child to an adult. That is because of mitosis. So mitosis is completed in four phases. You have prophase, metaphase, anaphase, and telophase. First, we're going to talk about prophase. In prophase, you're going to have all that long, stringy DNA. The chromatin condense into a chromosome, and this chromosome has two sister chromatids. So you're going to have this chromatid, and they're called sister chromatids, and they're going to meet at the center at a centromere. So you're going to have that happen, and then you're going to have this the nucleus start to disintegrate, and you're going to have all those chromosomes start moving to opposite ends of the cells in order to divide. So after all that, you then have metaphase. Metaphase is where all the chromosomes meet in the middle of the cell. And you can see how all these little dark spots right here in the middle of the cell are lined up in a complete straight line. Now, this is going to happen differently in both plants and animals because plants lack what is called a central. And for animals, the central helps create these spindle apparatuses, and these spindle apparatuses connect to the centromeres of these chromosomes, which help it to meet in the middle and then eventually spread apart. However, this plant cell, like I said, lacks that central, but it still has spindle apparatus. It is unknown how that spindle apparatus comes about, but plants do possess it. So after metaphase, we have anaphase, where these chromosomes split apart and they head to opposite ends of the cells. And like I said last time, they do this through the spindle apparatuses. And as you can see, all these dark spots right here, and how they're splitting apart in both this plant and this animal cell. So after anaphase, you have telophase. And within telophase, you're going to have two nucleuses forming around your two new cells. So you're going to have that happen. And you're going to have all those chromosomes turn back into chromatin, which is the uncoiled, stringy DNA in both plants and animals. So after telophase, mitosis is completed, and you move on to cytokinesis, which is the splitting of the cytoplasm. Now, this is going to be different in both plants and animals because plants have a cell wall, and that cell wall is not going to budge because it's strong. So it has to create a new cell wall, and it does this through a variety of steps in order to split. The animal cell is going to create a cleavage furrow, and this cleavage furrow is basically going to pinch them apart until they split into the two new cells. So after cytokinesis is completed, you finally have completed the process of the cell cycle, and now we can move on to meiosis. Meiosis is a cellular division that gives us our gametes. So meiosis takes place in sexually reproducing organisms and gives us the diversity we see today. So how does meiosis work? So meiosis, the cell is going to be divided twice. We're going to have meiosis 1 and we're going to have meiosis 2. So the first step is interphase. You're going to have all that chromatin that's going to replicate through DNA replication that leads you to prophase. During prophase, you're going to have these homologous chromosomes pair up together, and they're going to perform what you call crossing over. They're going to swap genetic material. Then after all that happens, we're going to move on to metaphase, to where these homologous chromosomes center right in the middle of the cell. Then after that, you move on to anaphase, where these chromosomes split apart. They move to opposite ends of the cell. And then telophase, where the nucleus starts forming again and the cells split apart using cytokinesis. And that is the end of meiosis 1. In meiosis 2, we're going to start out with prophase, to where the chromatin turns into chromosomes. And then these chromosomes are going to line up in the middle of the cell during metaphase, and then they're going to split apart in anaphase, and then the cells are going to divide, turn back into chromatin during telophase, and you're going to have 4 haploid cells result from meiosis 2.